let us discuss about Krebs cycle. So in the aerobic respiration, the third step is Krebs cycle. In glycolysis, the end product is pyruvic acid enters into mitochondria. Within the mitochondrial matrix, the pyruvic acid is converting into acetyl coenzyme A. That is the end product of second phase. And here in the third step, the acetyl coenzyme A, which is the end product of second phase, it is involving in the Krebs cycle. So acetyl coenzyme A, it is combining with the oxaloacetic acid. Here oxaloacetic acid is a 4-carbonous component. Acetyl coenzyme A, it is a 2-carbonous component. 4 plus 2, 6-carbonous citric acid is produced. Citric acid. Here acetyl coenzyme A is here it is cleaving or it is splitting into coenzyme A and acetyl coenzyme A and oxaloacetic acid. Both are combining and coenzyme A is separated and citric acid is formed. So the first produced stable compound, citric acid. So 6-carbonous component, citric acid. So the Krebs cycle is also called as citric acid cycle. It is also called as citric acid cycle. Krebs cycle is also called as citric acid cycle. And why it is called as Krebs cycle first? The total intermediate products are here observed by Hans Krebs, scientist. For this discovery he got Nobel Prize also. So here Krebs cycle, in the Krebs cycle the first stable compound is citric acid, that's why it is known as citric acid cycle. In the presence of citric synthase enzyme, so the first enzyme is citric synthase enzyme. In the presence of citric synthase enzyme, here addition of water, hydration process. So by the addition of water and acetyl coenzyme A and oxaloacetic acid, so they are forming the citric acid. Next, citric acid is having 3 carboxylic groups. That's why it is also called as tricarboxylic acid cycle. Citric acid is also called, here Krebs cycle is also called as TCA cycle, tricarboxylic acid. Citric acid is also known as tricarboxylic acid. So having 3 carboxylic groups. 3 carboxylic groups. So tricarboxylic acid cycle. Next step is, next second reaction is here dehydration process, removal of water. So this removal of water, it is catalyzed by aconitase enzyme. Aconitase. And again here after the removal of water, the produced compound is cis-aconitic acid. It is also 6-carbonous component. Next the third step is hydration process. Hydration, addition of water. Due to the addition of water, here again same component is produced. A removal of water, addition of water. So same component, citric acid is produced. It is also called as here isocitric acid. Iso means same type of citric acid is produced. A removal of water and addition of water. Isocitric acid. So the 2 second and third steps are catalyzed by the same enzyme, aconitase enzyme. And isocitric acid is undergoing one oxidation process. So it is in Krebs cycle, it is the first oxidation. First oxidation is taking place in this between the isocitric acid and oxalosuccinic acid. So isocitric acid is undergoing one oxidation. Oxidation means here removal of hydrogens. Hydrogens are removed from isocitric acid and these hydrogens are received by NAD plus. So NAD is taking hydrogens from isocitric acid. So here it is oxidized, it is reduced. It is reduced into NADH2. Two molecules. These are two molecules only. Two citric acid molecules, two cis aconitic acid, two isocitric acid molecules. Next, two oxalosuccinic acid molecules. So all are two molecules only. Starting number is acetyl. If one acetyl co-NMA is involving, all are only 1 1. Here two acetyl co-NMA molecules are involving. So here two cycles. Here in this between these two components, one oxidation. First oxidation is taking place in the Krebs cycle. So two NADH2s are produced. So this reaction is catalyzed by isocitric dehydrogenase. Isocitric dehydrogenase. Next, oxalosuccinic. The product is oxalosuccinic acid. It is undergoing one decarboxylation reaction. Here two decarboxylation reactions are taking place here and here also. So first, oxalosuccinic acid. Here two from this component carbon dioxides are removed. So after the removal of CO2s, it is converting into alpha ketoglutaric acid. So this decarboxylation reaction, it is catalyzed by oxalosuccinic decarboxylase. The enzyme involving in this step oxalosuccinic decarboxylase enzyme. Here after the removal of here up to citric acid to citric acid to oxalosuccinic acid, all are hexoses, are 6 carbonous components. Next, here one carbon dioxide is removed. One decarboxylation reaction is taking place. So 6 minus 1, 5 carbonous component is produced. 5 carbonous component. In this Krebs cycle, the first 5 carbonous, one and only 5 carbonous component is alpha ketoglutaric acid. This step is catalyzed by oxalosuccinic decarboxylase enzyme. So alpha ketoglutaric acid, it is also involving in the synthesis of amino acids. So here it is involving in the catabolic process, sometimes in anabolic process also, for the synthesis of some amino acids, for the synthesis of different products it is involving. So alpha ketoglutaric acid, it is undergoing one more decarboxylation. So addition of coenzyme A and removal of carbon dioxide. So decarboxylation process is taking place. Next, here one dehydrogenation also. Two reactions. Here oxidation means dehydrogenation, removal of hydrogens and removal of carbon dioxide. So these two steps are two types of reactions are taking place in this only. Dehydrogenation. From this hydrogens are removed. The removed hydrogens are accepted by NAD. It is converting into NADH2. So it is oxidized, it is reduced. Next, decarboxylation. From this component here carbon dioxides are removed. So decarboxylation. So dehydrogenation and decarboxylation. It is called as oxidative decarboxylation. So in the second step also, pyruvic acid oxidative decarboxylation. So decarboxylation, removal of carbon dioxides and removal of hydrogens. Here also same, oxidative decarboxylation, undergoing oxidation and decarboxylation. So it is the second oxidation reaction. In Krebs cycle, within the Krebs cycle, it is second oxidation reaction. So this reaction is catalyzed by alpha ketoglutaric dehydrogenase. So after this reaction, the succinyl coenzyme A is produced. Two molecules of succinyl coenzyme A. Succinyl coenzyme A, it is a 4 carbonous component. Why? It is 5 carbonous component. One carbon dioxide is removed. After the removal of one CO2, here 4 carbonous succinyl coenzyme A is produced. Succinyl coenzyme A, it is undergoing one splitting or one cleavage reaction. So coenzyme A here added, here removed. Next, one water molecule also removed here. One water molecule also added, hydration process. And here one GTP. So what is GTP? Guanosine triphosphate, like ATP. It is equal to ATP. Guanosine triphosphate. So here the GTP is produced directly. Here GTP, it is produced directly. It is also called as substrate level phosphorylation. Substrate level phosphorylation or direct level phosphorylation. As we discussed in the glycolysis, in the glycolysis also at 2 points substrate level phosphorylations are taking place. Here at one point, here GTPs are produced directly or by substrate level phosphorylation. So this reaction is catalyzed by succinate kinase or succinyl thiokinase. Succinate kinase, succinate thiokinase. Here 2 GTPs are produced by substrate level phosphorylation. Next, 2 succinic acids are produced. It is 4 carbonous component. It is also 4 carbonous component. Here succinic acids are 4 carbonous component. Next, all these are 4 carbonous components only. So succinic acid is undergoing one more oxidation reaction, third oxidation reaction here. Third oxidation. So in this oxidation, hydrogens are removed and received by FAD, flavoadenine dinucleotide. In the glycolysis or in the second step, and in Krebs cycle also in these points, here NAD is receiving the hydrogens. From this component it is receiving hydrogens, and here also it is receiving hydrogens from this. But here the hydrogens are received by FAD, not by NAD. Here FAD, flavoadenine dinucleotide, is reduced into FADH2. Next, this reaction is catalyzed by here succinic dehydrogenase. Succinic dehydrogenase. The next product is fumaric acid. In this reaction, fumaric acid is produced 4 carbonous component, and here hydration step, water molecule is added. So when water is added, it is converting into malic acid. So this reaction is catalyzed by fumarase enzyme. Fumarase. So malic acid is undergoing the last oxidation reaction, 4th oxidation. Here from this here hydrogens are removed, received by NAD. NAD is converting into NADH2. Here at first, second and 4th oxidation steps, here NADH2s are produced. If 2 acetyl coenzymes are involving, so all are 2. Here 2 NADH2s in the first oxidation, in the second oxidation 2 NADH2s, in the 4th oxidation also 2 NADH2s. But in the 3rd oxidation, FADH2s are produced. Here first and second, 4th oxidation reactions, NADH2s are produced, 6 NADH2s for 2 acetyl coenzymes, and 2 FADH2s are produced, and 2 GTPs. So in the Krebs cycle, only in the Krebs cycle is taking place in the mitochondrial matrix. The starting or the first stable compound is citric acid. It is also known as citric acid cycle, and tricarboxylic acid cycle, as it is having 3 carboxylic groups, and Krebs cycle. Here after the scientist's name, it is also called as Krebs cycle. And in this only Krebs cycle, total 4 oxidation reactions and 2 decarboxylation reactions. Here and here 2 decarboxylation reactions. At 4 points oxidation reactions are taking place. But in 3 points only, first and second, fourth oxidation reactions, NADH2, totally 6 NADH2s and 2 FADH2s, and here 2 GTPs are produced. So in Krebs cycle, the end products are 6 NADH2s, 2 FADH2s, 2 GTPs and 4 CO2s. Here 2 CO2s and here 2 CO2s. And the last enzyme is malic dehydrogenase. So these are the enzymes involving in the different steps, and the end products are 6 NADH2s, 2 FADH2s, 2 GTPs, 4 CO2s. It is about Krebs cycle.